It's my pleasure to introduce George Gordon, who has been working on endoscopic imaging and used ultrasensing techniques before, and will talk to us more about this today. So, George.
Yeah, thanks. So, so I'm in the from the Department of Engineering. So I'm still not quite at the medical coalface, so to speak. But I think what we do is fairly applied, and our main application is to try and make better endoscopes. So the main motivation for my particular work is esophageal cancer. So esophageal cancer is a major killer, and what will typically happen is over time people might get some acid reflux in their esophagus, and they might go and see their GP, and then they'll be referred for an endoscopy. And the endoscopist will use this instrument called an endoscope, which is basically like a camera that's put inside your esophagus, and they'll look at the images to try and follow the progression of the disease. So on the top here we've got like a healthy esophagus, and then below that someone that's had acid reflux over time gets this disease called Barrett's esophagus, and you can see these discoloured patches here. It's quite easy to diagnose this disease. But the difficulty is that of these people with Barrett's esophagus, about 1 or 2% will go into develop dysplasia, and which is like an early form of cancer, and then they'll go into develop esophageal cancer, which is very deadly. And the difficulty is that using these conventional endoscopes, you can't really see which of these people with Barrett's are likely to progress into this. So basically, you'll be monitoring them, and then one day you'll come in and you'll monitor them, and you'll see that they've got already quite an advanced stage of the esophageal cancer. So part of the problem is that these white light endoscopes, when they're used diagnostically, they actually throw away a lot of the information that you can get from light, and so they can't actually see these early stage changes. So one kind of obvious way they do this is that they shine white light in, but they only detect light coming back in 3 wavelengths of light: red, green and blue, which is what your eyes do, but this is a fairly arbitrary choice of wavelengths for looking inside the esophagus. And these other wavelengths that aren't typically used can show you important information. So for example, if you look at people's skin under UV illumination, you can see damage to the skin much more clearly than you can with visible light. And there are many examples in nature of patterns on flowers, for example, that are only visible with ultraviolet light and we can't see with our eyes. A second property of light that is discarded by conventional endoscopes is optical phase. So if you consider that light to be a wave, an electric field wave, normal cameras, they measure the power of the wave at different pixels, but one thing they don't measure is the offset between the waves at different pixels, which we call the optical phase. And you can think of this as being a bit like when you look at a sort of a degraded old tombstone and you can't see what it says, but if you can access the depth information by, for example, doing a rubbing, you can read it. And this kind of aspect of optical phase is used widely in microscopy, in phase contrast microscopy, and it greatly improves the contrast of cells in sort of a liquid solution. So there's a lot of extra information there that we can't normally see. And the third property of light that is thrown away by most conventional endoscopes is polarization. So again, considering light as a wave, electric field, it can oscillate in one of 2 axes, and we don't really record that information in conventional endoscopes. And this can be very useful. So in the natural world, for example, if you look at things like reflections off bodies of water, that is quite different when you look at different polarizations of light. And also there have been numerous studies that show if you look at the polarization properties of tissue, you can see things that you can't see with normal unpolarized light, but you can see these differences happening when you look at polarization properties. So there's kind of a wealth of information there that is discarded by conventional endoscopes. And the group that I work in, led by Sarah Bondique, we're actually looking at making endoscopes that measure all these different properties. So we've got the wavelength, the phase, and the polarization. And so within that research group, my particular work is on developing endoscopes that measure phase and polarization. And there's a poster later on by a colleague of mine who's going to be talking more about the hyperspectral, the multispectral aspect. Yeah, so other than being able to detect these additional things about tissue, there are some other key advantages of detecting optical phase. One is that you don't need to use lenses. So I'm making a fiber style endoscope, so you can just image using just the end of the fiber. You don't need any other optics. And that's good because it means you can electronically readjust the focus. So you can imagine, for example, if you were using this, what you could do is you could sort of insert the endoscope into the esophagus, and you could take a sort of wide field view and see where cancers are, and then by pressing the endoscope against the target area, you could then see at a microscopic scale what's going on, and that opens up potentials for things such as optical biopsies. And another key advantage of this is because it doesn't require any additional optics, the endoscope need only be as thick as the fiber, which is typically of the order of about 400 microns in diameter. So you could fit these inside a needle, and you could actually minimally invasively use these to image areas of the body that are typically inaccessible to conventional endoscopes such as the brain. So that's kind of a sort of wider future aim. And this is kind of a more immediate benefit of it. So how does this relate to maths, which is why we're all here? Well, actually, I mean, effectively it's all maths. So we're trying to measure this quantity optical phase, but there's not really such a thing as a phase sensor. It's always typically has to be made from multiple other components. So we're using just normal cameras, and the way we use the phase is through these what we call phase retrieval algorithms. So that's one way. Secondly, because we want to make this work properly, you have to slightly change the way you spatially illuminate things, and similarly you want to do spatial filtering on the way back. And to do that we use digital holograms, which is another quite involved field of maths. So once we've been able to control these light fields and their shape, we then have to send stuff down the fiber and get it back again, and it gets all scrambled, and so we have to undo that scrambling. And then once we've unscrambled this light, we then have these raw measurements, and we want to infer from those some particular physical properties of the tissue that aren't immediately apparent in the measurements. So we apply Bayesian inference techniques to figure out those properties. And then a broader thing that we haven't really done much on yet, but we will be doing as we collect data, is how do we actually take these huge multi-dimensional data sets with all these wavelengths, phase, polarization, and actually link those to histopathology so that we can actually grade cancers. And so today I'm going to talk about the collaboration I've had here with Milana and Francesco and Anders Hansen, and it's talking about this particular aspect, which is unscrambling. So just to explain in more detail about that, what happens really is ideally you have this imaging fiber here, and it's made of lots of little bits of glass fiber always kind of sandwiched together in this kind of huge honeycomb thing. And ideally you should be able to send an image down it, but that's not the reality of how it works. What really happens is, because of bending and because of changes in temperature and so on and so forth, actually what comes out is quite scrambled, particularly with regards to phase and polarization. So we actually need to compensate for that. And this is where the maths comes in that I've been working on with Milana. So we model this particular problem as being a linear inverse problem. So I'm just going to run through sort of some basics of that. So we imagine that what we're doing is we're sending in some optical field into the fiber, and what comes out is some other scrambled optical field, and the link between these two is defined by this function here, which is a point spread function. So basically it defines how one single point of light here, what field that produces at the output. And if we sum all those together like this, we get this convolution integral, and we can actually calculate for any given input what the output is going to be. So that's the kind of model of how this works. But that's only a really small part of the problem, actually, because in reality this is not a continuous We can't treat this continuously because we have to use a camera that only has finite pixels on it, and similarly this fibre here only has finite points at which it samples the incoming light. So we actually have to discretize this problem. So we turn this field here into a vector of points, column vector of points, and similarly the input, we'd also turn that into a column vector of points, and then this convolution integral then becomes a huge matrix which maps every point at the input to every sample point at the output. And then the goal is to try and recover this huge matrix so that we can actually undo this scrambling. So I'm just expanding that out just to sort of demonstrate a point. You don't need to read every single number on there. So yeah, so just for example, if we send in some input field here, which is represented by one column times this matrix, comes out to an output column. But of course, that in itself is not enough. You can't just do that and find out the matrix, because this is the thing we don't know. We know this, we know this. So what you actually have to do is you have to send down multiple different inputs and measure multiple different outputs. And as you do more and more of these, you can actually start to get a reasonable approximation of what this is here, because it becomes more and more constrained. So the idea is that we eventually want to figure this out. So it's actually solving many, many inverse problems to eventually find this. And this is where the work that Milana was talking about comes in, because we want to do this, but we can't just take, in this case, if we wanted to, we could, in theory, just take enough measurements such that these were both the same size as this, and then we just do an inverse. But that would take a very long time, and the patient can't sit there for 4 hours while you're measuring that. So what we actually want to do is we want to minimize the number of measurements we take. And so this is an important aspect of where the compressed sensing comes in. How can we figure out ways of taking the minimum number of measurements? And also there are some very promising Bayesian approaches to try and make the most of the data we have, to try and converge something as quickly as possible. And the second thing is also the endoscopists will want to see the results as quickly as possible. And so we have to be able to process this data very quickly. And it's quite a slow process at the moment, because you might have a huge matrix and you have to solve for every column. However, I've been working with Milana and Francesco here about trying to, as Milana said, use a different sampling basis. So instead of just using the pixels off the camera as our sampling basis, can we think of a better function to use as a basis such that the problem becomes much quicker to solve? And we've got some promising avenues there which we hope to look into. And of course, underlying all this is the fact that we can use these various approaches to improve the speed, but the quality also has to be sufficiently good for medical diagnosis. So at the moment, we've kind of got this fairly naive approach working, which is fairly brute force minimization approach to solving this problem. So this is just to show you what we send into the fiber. It's some light field that has certain amplitude, phase, and polarization properties, and then it gets scrambled, as you can see, quite badly, particularly in the phase and polarization. And then by applying this sort of inverse method, we can get a fairly good reconstruction of what we sent in. But currently this is really slow, and it probably takes us about an hour to get enough measurements to do this, which is too long for using inside a patient, and it takes us even longer. It takes, well, I don't even know if I should say. It's embarrassing how long it takes at the moment. But we're aiming to reduce the processing time to actually reconstruct this to be something of the order of minutes rather than hours, so that we can get some data. Yeah, and so working with my collaborators in the Department of Oncology and Physics of Medicine, we also do have some. We've tried this on some biological samples, but I'm not presenting those yet. Partly because we've not published them, and also partly because we don't really know how to interpret what's come out of this, because it's quite an unusual modality of looking at samples. Yes, so yeah, I'd just like to finish by thanking everybody who work with and provides all the samples and the maths. Yeah, and I'll hand over to Bogdan to deal with questions. Thank you.
questions? Yes, in the back.
So two questions. So you seem to be headed for label-free detection based on tissue autofluorescence and spectral imaging of that autofluorescence. But is there any evidence on tissue sections that you can detect early dysplasia in that way?
Yeah, so I didn't quite present the full picture. So the hyperspectral stuff, which is an aspect of this, is actually looking at using hyperspectral techniques to use multiple fluorescent dyes together and try and decouple the effects of different ones. So we are looking at that too. But the phase and polarization stuff is label free, and there has been some work like phase contrast microscopy and polarization polarimetry to show that the microstructure, the disordered microstructure of early cancer does scatter the phase and polarization differently. But what we don't know yet is how sensitive that is to different grades. So we know that, if it's a sort of reasonable sized lesion, that it does scatter light differently than the surrounding tissue just from microscopy studies, but we don't know exactly how sensitive it is, and that's something we'll have to figure out. Hopefully it's reasonable.
And also your image reconstruction time is, if you're going to use it with endoscopy, it's got to be real time.
Yeah, so this is the thing. So this method I showed here, was just something I kind of cobbled together. That I'm not a mathematician, so I'm hopeful that some of these methods will be, very fast. So hopefully in the order of minutes. I guess one way of getting around that is that, particularly if we use some of these.
Minutes isn't going to be fast enough. It has to be real time if you're going to use it with endoscopy.
Well, yeah. So I guess one approach to that is that you can actually see. So the amplitude information is not that corrupted. It's only like a little bit corrupted. So you can actually. The idea is to one thing we could do is send down normal kind of broadband white light, and you get a reasonable image to sort of so you can see broadly. And then if there's an area, perhaps you can sort of, I guess, take effectively an optical biopsy. So you can maybe just switch on this feature at a particular point, perhaps, is one way of sort of doing it if the speed is too slow, and then sort of.
But then you're selecting on the basis of white light illumination, which we already know is not very good.
Yeah. So I guess what remains to be seen ultimately is what mixture of these different features will work. So it might be that the hyperspectral aspect combined with dyes is a good way to find the target region, and we might find that the phase is a better way to actually do optical biopsy. So maybe in the end that we actually have all these things in one endoscope and we switch between them for the different modes. Because you're right, it is potentially the speed is going to be a major issue. To be honest, right now I don't know how quick we can get it. It might be that we can get it down to sort of like a sort of seconds speed. I'm hopeful. Yeah.
Okay, so a lot of challenges. It's a good thing for researchers.
About how much data are you talking about gigabyte-wise that you're collecting for the standard study?
So at the moment, because of the resolution of if I collect a 2 mil by 2 mil area, the amount of data I need to reconstruct all this is probably about maybe 50 or 60 gigabytes of data, but it's not really. That's not necessarily representative of the amount of information, because I'm actually using really highly uncompressed images to get really big dynamic range. So I imagine, probably if we were to do this, develop it further, we could probably shrink that down quite a lot.
You could distribute it across a large number of computers as well for handling things in parallel.
Yeah, it should be, because we analyze the different regions separately. Yeah.
In the back. Yeah.
Back to the speed question. So, what kind of hardware software environment are you using to achieve this calculation? And do you think that making that bigger and better could help you get the limit down to real time or close to real time?
Yes, I think definitely. So each of these, the problem is we've got to solve loads of inverse problems, and maybe thousands and thousands. And the problem is we can only solve on one computer a certain number at a time in parallel. So it is highly parallelizable. So that's why I'm hopeful that we can do it in kind of in real time. So at the moment, I'm just using, I'm an engineer, so I just default to the usual MATLAB, which is sufficient. But I imagine if we did it on if we do a GPU implementation, it should be a bit faster. There's also some inference stuff we use for the polarization things I don't really talk about. That's quite slow too, because it's using this kind of weird framework though. It's not really well adopted.
Can I just clarify? This matrix D that you're estimating, to what extent can you estimate it once for a bundle of fibres, and to what extent does it need to be estimated every time you put it in the patient?
Yeah.
So they're obviously two very, very different problems.
Yeah. So I think that's something that we're going to have to study, and is going to probably play a major part in getting the speed down. And what I suspect is that there'll be the matrix will have certain statistical properties that are always preserved. Probably the amplitude of coefficients will be fairly stable. What it will be is sort of a variation in the phase between will probably be the thing that changes. So that's one thing we're looking at. If we know that there's like a sort of a Gaussian sort of link between different parameters of it, then hopefully we can use that as a starting point and very, very quickly converge to just the phase values.
You're more or less solving a blind deconvolution problem. It's just you have multiple T0s you can take advantage of, right? So your H is unknown, and it's also changing spatially. So
Yeah.
what are your assumptions on H that you put into the reconstruction?
The assumptions are that it is. I don't know how to express it in terms of H, but when we discretize it, that matrix D, we're assuming that it's sparse. So that, which I guess in terms of H means that it starts at a point here and it doesn't really extend very far. So if you've got a huge field here, it only really spreads a small amount.
So Van diagonal.
It's like diagonal. We haven't actually been able to diagonalize the matrix because of the basis I'm currently using. But it's effectively sparse in some kind of sense. Yeah.
I had a question which was very much in line with the measurement. So one of my older collaborations, we looked at cable communications in a similar setting, and there is interference between the wires. So I'm not sure, is there interference between the fibers and the cable as well that produces? Yeah, so it's not just a bending. And you can. So what they were doing is they were using compressed sensing techniques to account for the interference, and that was their matrix. How much do you think you can use measurements that you take once or a few times in a static context for your whole cable of fibers, and then with that information improve on the estimation later on? And that's where probably you can use learning things as well. Is there something that you have considered, or?
I think the answer is a lot. Like people have said, that's one thing is you can map the statistical properties, but there's also lots of other low-hanging fruit. Like I'm currently using a megapixel image of the output of the fiber, but actually you're right. It's the only thing that matters is the coupling between these individual little fiberlets inside bigger fiber, and it's only about 5,000 or 6,000 of those. So actually, even though I'm using like 1 million pixels, I should be able to hugely reduce that down to maybe only like sort of 5,000 by 5,000 matrix or something, which is much easier to deal with.
But in their context, the cable is fixed, so they don't have a problem. In your case, cable moves.
Yeah.
And the bending changes, so you're. But there is, I suppose, information that at the core should provide an edge or a better starting point.
Yeah, well, the length of the fiber is fixed, and the bending is never really that sharp. So you do expect that like light from this one will couple to the next fiber, and then a little bit to the next fiber. By the time you get sort of 3 or 4 fibers away, over that distance, you get almost no coupling. So that's the kind of key, I think, to doing it quickly.
Okay. If there are no more questions, then I'd like us to thank the speakers again. And